Welcome to our podcast, Biotech CEOs. Today, we're going to do something special. We're revisiting some of the most compelling stories and CEOs of our earlier editions to update their journeys. I wanted to do this for two reasons. First, because as I've said so many times before on these podcasts, I love the people in the industry. They're among the grittiest, most determined, and above all, most philanthropic in terms of their commitment to their science and patients, people I've ever known. It's often said that opposites attract, and coming from the perspective of an investment professional, where essentially everything I do on a daily basis is about money, these are generally people who don't care about money at all, except as it's necessary to move their science forward. But the second reason is less personal, but definitely more important. By following these individual journeys, I hope somehow to demonstrate how incredibly fast all of this innovation is taking place. Of course, this was demonstrated back in the height of the pandemic when Moderna and BioNTech, working with Pfizer, were able to create vaccines literally days after the COVID virus was genetically sequenced in China. But now that the virus has receded, at least in the US, and vaccine boosters are taken for granted, we've forgotten how incredible that formulation was. Anyway, I've been thinking about a way to viscerally demonstrate the speed at which this change is occurring when I happened to walk into a high school history classroom at a private school I'd been considering for my grandson. I've always loved history, but I was taking this adult seminar more to check out the school than for any other reason. As it turned out, I discovered that the teacher, Dr. Nick Hoffman, had actually done his PhD on Civil War medicine. That's right, the Civil War, when the treatment was generally off with their limbs, in other words, amputation. And it hit me, what better way to demonstrate the speed of innovation than to juxtapose it with a period when medicine was so primitive? So Dr. Nick Hoffman was our guest in May. Scheduling issues kept us from directly following up that Civil War focused podcast with this follow-up edition. Nevertheless, now that we've been able to reconnect with some of our favorite founders from earlier issues of the podcast, we hope you'll really enjoy re-engaging with them as well, with the challenges of medicine during the Civil War era in your mind's eye. Abdulkader Rahmo joined us in August of last year from SMS Biotech, a San Diego company using small mobile stem cells to attempt to regenerate respiratory air sacs for patients with COPD.
Now, right now, we finished the correspondence, the pre-IND with the FDA. We know now what type of test we need to do. The FDA wanted us to go for human testing on patients, not on healthy volunteers, which we welcome actually, because that might potentially give us early signs of efficacy during the early phases of clinical trial, which is the phase 1 trial. So otherwise, of course, we have to go through the standard GLP tests, bench test, and others to be ready for our IND submission. Which to that goal, we are working quite strongly with all the means that we have. And we're hopeful that soon we'll go to clinical trial and we will benefit people. Now, we do intend also to include a compassionate use once the FDA approves it, which could occur after safety data, some safety data on a human. And that means people with severe COPD could have access to this remedy early on. We don't have to wait until phase 3 or after commercialization for making this available to people. Saving lives is a privilege, and it's something that we all very proud about. And we intend to work very hard to make that possible. Yes.
I'm very excited to hear that, AB, especially since, as you know, from our earlier conversations, why my father passed away as a result of COPD. I've been an asthmatic and all that sort of thing my entire life. So I may be first in line with you. So that's really very exciting. So when do you think you might actually try it on that one individual?
Yeah, I mean, we are hopeful that we be able to submit the IND within month. And I have to state the date. I mean, do I have to state the date?
No,
or will be yeah.
No, of course not. Of course not.
Yeah. Yeah, but it's also dependent, on satisfying the FDA with all the requirements. Usually, after submission, there is a short period, about a month, for the FDA to review the material. If they are happy with it, then we can definitely go into clinical trial. Now, we are planning on preparing for the clinical trial, and assuming everything will go as planned, of course. And I think it's not just us, but also the FDA, the NIH, all institutions will work hard in making or presenting an alternative for COPD patients. So I see that as a partnership towards a goal. I understand fully that we need to fulfill all the safety requirements, but I'm extremely optimistic about the safety issue. We've been through many tests with the animals. We've been discussing it with physicians, with scientists, and we are extremely lucky, I think, to have an agent that is very safe and apparently present no danger whatsoever. Now, we've seen in the market some willingness to risk for patients who are desperate, willingness to take some risk with certain cells that are dangerous. Now, our advantage is, I guess, definitely is we're going to provide this to patients who are, although willing to risk certain danger, do not have to.
Do not have to.
Yeah, they do not have to take any risks. We will be happy to present the cells in what as safe in large quantities, introduced directly to the lung via nebulizer, and benefiting from all these great advantages these very unusual cells provide.
So now, but I think that, as I recall, when you did tests originally on mice with the SMS cells, you injected the mice. Is that correct?
We started actually with rats, but we included later also mice.
Okay.
And we tested all kinds of quantities. We injected them systemically. We injected them directly to the lung at various large concentrations, various recipes. We didn't see any adverse reactions so far. And noting also that we lately used immunocompromised animals, not just healthy animals. So either healthy or immunocompromised. None of these animals had any adverse reactions that we can detect towards these cells, which are human. Now, these cells are the human, of course, stem
Right.
cells.
Would you anticipate any differential in efficacy resulting from the difference in delivery between injection and a nebulizer?
Yes, I do. I think definitely presenting the cells directly to the lung is of a great advantage. And it never has been achieved with other cells. So we think that nebulizing and directly submitting the cells to the lung will be of big advantage. Now, the fact that we did it to the animals this way, and we're gonna do it the same way for the human, helps with the translation. You know, we're trying our best to mimic the experiments and mirror the experiments on the animals on what we're gonna do with the human, vice versa, so that. For example, we're going to use very similar technology in measuring success in human to that we are using in animals. So this will help in the translation, and that will help us gain confidence in that aspect. You know, translation from animal to human has been not so efficient whatsoever. So we need to be careful about pushing every edge towards towards ensuring success by mirroring the technology and the application that we apply to animals into human.
What would make you really happy and excited in terms of results?
What will make me happy about the results is what having the patients being happy, you know. So it's about the patient deciding that this really changed the quality of his life. And I have to say, I mean, most severe patients have really modest requirements, you know, modest expectations. They have their ambitions have been dampened by the inefficiency of the current medicine.
Right.
So I think if we can present to them something that satisfies them, we'd be extremely happy. Now, of course, patient will be looking for more and maybe more improvements, and we hope that we can improve the recipe and achieve what we have achieved in animals and cure this patient from this disease. I think it's doable. I don't think it's science fiction. I think we can achieve that, and we need just to galvanize all support and the bright minds behind it. And it's not just science; it's also business, and it's also you know, all what it takes to make a company successful.
AB, you said when we spoke last that you know, you hadn't yet published on SMS cells. Does this put you in a position where you might be more willing to do that as you get into a phase one, or would that come later? How would that?
Yeah, we had limited publications, but we published 10 patents.
Right.
And we didn't publish much in journals so far at all. I mean, I had only one publication, but Uh, the publication is definitely something we intend to do. We think that we might be at the verge of doing other applications and testing the system, the technology on other applications. For that, we need a broad support, a strong support, because investing, in these different applications is not going to be a trivial amount. If we can convince the people through publications, that's fine. If it takes publication to do that, I have no problem with that. I think for us, publication uh, is secondary to achieving good results with the human. The key is for us to the patient to achieve something for the patients. Once we achieve something for patients, I think publication will be a very easy step, and we will be very well received, and it will be helpful in spreading this technology.
Well, AB, I'm very excited to hear the news.
Thank you.
I'm excited for you because I think you're a terrific person, very generous with your time and your intellect in terms of helping mankind.
Well, I think it's a team effort, and I'm proud to be part of this team effort. And if any prize will help our mission forward, that I'd be happy to get that prize. But the important thing in my mind is really the prize of helping the patients and achieving what this technology can achieve, its utmost potential. And we will get there with the help of people like you, supporters who believe in us and who stimulate us continuously. Thank you.
Well, thank you, AB. I so appreciate your time today. Torrey Smith was our guest a little more than a year ago on a podcast entitled From a Dirt Floor in the Sierra Nevadas to the Biotech Shark Tank. Torrey is the CEO of Endiotics, a company which is developing a small pill bot for use initially as an early, low-cost, low-stress kind of diagnostic tool in gastrointestinal diseases. Torrey, let's first remind listeners how the pill bot might work and how it could benefit the healthcare system and patients.
Absolutely. So PillBot is basically a moving eyeball in the human stomach. And our goal is to enable a gastroenterologist to have a quick look around inside your stomach, but over a Zoom call from home. So this would sort of transform your hospital visit into a Zoom call for the upper endoscopy population.
Great. And last May, you had mentioned that you'd filed several and had been granted at least one patent. And various members of your team, including yourself, had swallowed the pill, I guess, 24 times at that point, really going from a kind of technology demonstrator to something approaching a viable product. But you thought you needed smaller motors, lighter batteries, that sort of thing. How's that process progressing?
So it's been a very exciting time. Basically, we have much smaller motors. We found batteries that are capable of putting out about 3 times as much electric current for the given size and weight. And so we're very excited to debut this technology at Digestive Disease Week in about 2 weeks. And so the company is just in a hardcore hardware and firmware and software sprint to make sure that we give people a fun demo because we're going to be doing 3-dimensional flights of these robots in our stomach models.
Wow, terrific. And so what is the process in terms of the FDA to use it in humans, human trials for those who aren't part of the management team?
Sure. So the first step for us is putting together a pre-sub meeting. We're hoping to actually meet with FDA in September to discuss the latest and greatest part of our tech, to sort of help identify what our path through the FDA is going to look like. Right now, it looks like there's a good chance we might be able to use what's called a predicate path, and sort of pay homage to the world of passive pill cameras and also to the world of magnetically actuated pill cameras. And so we're actually thinking that we might be able to be in the market treating patients in the 2026 timeframe.
Right. Okay, great. And so what are the next sort of steps that we might trace for Endiotics as we go through the rest of this year?
Sure. So we a few months back, we were able to announce that we had signed a know-how agreement with Mayo Clinic, which is a very important step in us engaging the great doctors of the world to take this, out of the R&D world and into proper clinical trials. And then we just got back from the Edison Game Changer Award, where the Thomas Edison Awards are calling PillBot a game-changing technology. So very excited. At this point, I would basically say that it looks like the world is asking us to make PillBot real, and now we have to deliver on that request. And so I'd say the next 3 to 6 months represent us fine-tuning the size and shape of the robot, making sure that our three-dimensional flight in the stomach is robust and reliable, and perhaps most importantly, getting our live video signal from just a few pixels and a couple of frames per second, where it's kind of more like a demonstration of a possibility. We need to get good, crisp, clear live video and make sure that we can take that into our first clinical trials.
So last May, Torrey, you said that you've been wishing your entire career to be in a situation where you had $3 million and a company worth at a metric of like 15 million at the time, with the goal of building a company to $100 million. But that at least last May, you found the prospect terrifying. How are you feeling these days?
Well, we're very excited to be here. We just secured a $1.5 million commitment from the Verge Health Tech Fund based out of Singapore. We've been able to close half of that money, which oversubscribed our seed round. So we're very, very pleased to be able to officially say that phase of the company is complete. And now we start to see Series A on the horizon. But the fun thing about when people write you checks is that you then have to deliver on the reason why they wrote those checks. And so we are just nose to the grindstone, basically just getting ready to actually make this thing real.
I'm sure you are. Torrey, thank you so much for agreeing to spend just a few minutes of time with us today in what I know is a really busy schedule. But congratulations on your progress, and we'll be looking forward to additional milestones for Endiotics.
Tim, thank you so much. It's a pleasure.
Nikhil Shah joined us last fall in a podcast appropriately titled Grit is Everything. His company, Nephrodite, is working on a kidney replacement device nicknamed Holly in honor of a patient who struggled mightily with late-stage renal disease and finally succumbed to it. Nikhil, maybe you could start just by creating a little context here and reminding listeners of your vision for Holly and how it works.
Absolutely, and thanks for having me back. This is a real pleasure and a treat. There was no question in our minds that when dialysis was pioneered in the late 1940s and really took off in the 1950s, it was life-saving. It was offering patients the chance to keep living and be with loved ones and family, and to some degree be productive. But it was a tremendous burden, and that was the trade-off. What was surprising to us is that we're almost 80 years into it, and unlike our cell phones, computers, other things that we've done, we have not really innovated in the space of dialysis and possibly in renal care to a high level. And so what we wanted to do was ask patients, find out from our patients, our family members, the loved ones that were on dialysis and facing the challenges of end-stage renal disease. what is it that you would want in something that was progressive, something that would help you? And they identified 3 areas. They wanted a continuous device because they did not like the intermittency of treatment. They were challenged by their grafts and the access that they were having to use. And the third, and perhaps the most poignant, was that they lacked mobility, freedom, and independence, and that they would trade living for years on dialysis if they could just have 1 year where they were more free and more independent and more mobile. And so our device really took all of those things into consideration. And what we came up with was a hybrid device, an implantable device that would do the job of what our kidneys are supposed to do, a filtration device that's surgically implanted. And we coupled that with a wearable, either a backpack or a vest, that would allow patients the freedom to move around, go to work, birthday parties, to be with family. And at the same time, it removed all of that access issue. And so we're really excited about the device.
Okay. So you mentioned in our first interview how important it is for medtech entrepreneurs to approach the regulatory agencies early and often. And with Holly being an FDA Class 3 technology, why the requirements for you are especially rigorous. So I know you had applied for breakthrough designation with the FDA. Can you tell us where you stand on that and with the FDA as a whole?
Absolutely. So a couple of things on that. We applied for a breakthrough consideration with the FDA, and what we got was tremendous feedback. And what we heard from them was we weren't breakthrough ready, but we were definitely breakthrough worthy. And they gave us a list of things that we needed to prepare for. The second thing I will say is that it started a tremendous dialogue between us and the U.S. regulatory agency. And so because we are speaking to other regulatory bodies as well, and it began a great conversation for us in order to set the path forward in what we needed to do. And oftentimes what happens is when companies like ours building a complex device, there's unknowns and vagueness along the way that sort of really make it muddy and cloudy in terms of the path forward. And what I'm really excited about is there's an extreme clarity on our part in terms of the path forward, and a further engagement with the FDA, sort of an open door policy, given the fact that our device is addressing the needs of a class of citizens that are sort of really in trouble.
So tell us about what is the path now toward human implantation?
Absolutely. So what we have a strategy where we've been de-risking and doing the appropriate studies to get the device ready for its human grade sort of attributes. And we're near complete with that. We have some chronic animal testing that we're going to be doing, which means we're going to place our device in an animal, wake the animal up, and allow the animal to sort of move around and monitor the animal. That will then lead us to build with all the right components, the right coatings, the manufacturing capable internal mechanisms, so that we can get to a point where we land in our actually first in human trials. And so over the next 18 months or so, we're going to be building that human grade device.
And what sort of animals? Has the whole monkey issue been difficult for you, or?
So the best animal based on the literature for implantable devices, especially cardiovascular devices, is the sheep model, the ovine model. And so our early studies that were done in acute animals, so just 6 hours, they were all anesthetized. We typically do those in pigs. The pig is very resilient for short-term studies, but for the longer-term studies, so all the earlier studies in cardiac implantables, ECMO, that really became well known to people during COVID, the ECMO studies were all done in sheep, and they're very resilient for longer-term studies.
It's so important. So I'm grateful to you and to everyone like you for what you're trying to do good things for patients and, again, for our country. So thank you very much, Nikhil.
I appreciate that.
It's a pleasure speaking with you again. Rasab is the CEO of Cygenics, a company seeking to enable genetic sequencing at the local point of need in small batches and therefore with a quick turnaround, and at a price that would be reasonable to the common man, even in developing countries. One of the things that we're always talking about on the podcast is grit, the determination to succeed. And Tan, your past year is a huge indicator of that.
In the US, to do the next stage of our development would have required at least $5, maybe $7 million. We can do the same work here for approximately $2 million. So that's one of the main reasons we decided to do the next stage of our development from Malaysia, Singapore, and Taiwan. So even though it's been a very challenging, I would say almost 18 months, but the light at the end of the tunnel is getting bigger and bigger. So one of the biggest challenges in companies that are doing microfluidic-based devices is how do you make these microfluidic cartridges at a low cost, and they can be manufactured in high volume numbers. Where this has succeeded is those little, strips that you use to test your blood sugar. They're digital microfluidic devices. You have your little strip, you put it into your reader, and then you put your drop of blood in. The manufacturing of that has been perfected. They're still relatively expensive in comparison, compared to the normal paper strips that doctors sometimes use to check your urine or other samples. These strips are still, they're not cheap for the poorer people with lower incomes. But this is an example of a microfluidic device where the manufacturing challenge is a bit solved. Another area where microfluidic devices work is inkjet printers. So now you have a case where inkjet printers are actually very cheap, but the cartridges are very expensive.
Yeah.
But manufacturing of those in volumes has been solved. So one of the biggest challenges we had from both as a manufacturing process, as well as optimization on the costs, and also reliability and reproducibility, was a question which some investors were concerned about. So we've managed to solve this by actually adapting an existing technology which is used in LCD screen manufacturing. So now our microfluidic chips, the fundamental electronics or the arrays, we're going to use LCD TFT manufacturing technologies. So in other words, the foundries that make the electronics that go into LCD screen panels is called a thin film transistor arrays. So those same companies can actually make 90% of our product using the exact same processes. So, and TFT fabrication is pretty much standardized. There is innovation coming up, but we've been doing it for the last 30 years. And in the last 15 years, even the Walmart now sells you 50-inch screens for a couple of $100. And if you were to go to Alibaba or any sort of component website and you type in 4-inch TFT screen, if you want to buy 100 of them, you can get them for $5. So we are adapting technology from that, which will allow us to actually control droplets down to 0.5 millimeter. And our platform now becomes almost like a molecular canvas. So it's much more electronically controllable. The software can actually design the protocol processes for different chemistry processes for assays. It's much more software driven now. The hardware challenge has been solved. We're just now in the process of getting a few thousand of these cartridges made, and then actually start testing and fine tuning them. And once that's done, we know to get about 1 million of these done. Not 1 million, sorry, about 500,000 of these done costs about $2 million for us. And if I can actually do 500,000 tests a year, I'll be very happy, man. That will give us a company, a great advantage. So TFT manufacturing, the Far East, which means China, Korea, Japan, Taiwan dominates. They can make millions of these cartridges for us at a very low cost per cartridge, which basically solves one of our key challenges. And we don't have to reinvent manufacturing processes. We don't have to, create very expensive factories. So now we know that bit is sorted out. The next element is then integrate the system that we will have that will do the sample processing with the sequencer that we're going to use. And again, this part of the world, you have a lot of what I would call mechanical automation experts. Shenzhen, Taiwan, Korea, Japan, Singapore, Malaysia, all these countries have a lot of fabrication, mechanical engineering, integration experts, companies who can actually literally take your designs, get them ready for manufacturing relatively quickly, between 3 and 6 months. And within 6 months, you can actually start producing your, you can actually start rolling them out of the factory and delivering them to the customers. So even though it was a setback and all those issues that occurred, actually all those issues occurring actually benefited us. That delay, my inability to get things done in the last 6 months because of our feedback from investors and so on, actually has given us a better product, which is much more scalable, much more reliable. We can get manufactured at a lower cost and actually bring it to the market much faster. So there is a saying, in many religious cultures, that you may have a plan, but God has a better plan. So sometimes when things actually turn out to be completely against our plan, and we feel very stressed, we feel very anxious, the world is about to collapse, I would say sometimes it's ride that anxiety, ride that sort of destructive feeling out, carry on with what you're doing, because out of that, something better will come.
That is,
and I feel that's what happened to us.
Yeah, that's great advice for my grandson. I will pass it along. Thank you so much for spending some time with us to update us today, Tan. I've really enjoyed following up with you and getting to know you better. And we will, I'm sure, follow up again to hear more about moving into the clinic and that sort of thing. So we've got Jake Glanville with us now. He is the CEO of Centavax, a company that is focused on creating new universal vaccines for various diseases, first influenza. Jake, tell us, just kind of as a refresher, how do you actually go about making a universal vaccine?
Thanks for having me on, and good to talk to you again. Sure. So it's kind of like judo. We use the mutation of the virus against it. So in the case of flu and coronavirus and HIV and dengue and Ebola and a whole bunch of others, they get around our vaccines by mutating so that what you're injected with is no longer what you're going to be infected with. And most of your antibodies will not recognize the new version of the virus that's mutated. And we all know this. This is why they're updating those coronavirus vaccines now, and why they've updated flu vaccines since 1947.
Great. Well, the Centavax has had some interim successes since we spoke. I know that you're involved in raising a Series A to do some manufacturing. You're talking to some other people. Tell us update us on what's happened.
Yeah, so it's been
how are the pigs doing?
Been a good 8 months. Pigs are protected. What we've done in the last 8 months was since 2016, we've had in vivo data in pigs and ferrets and mice showing our platform delivers ultra broad spectrum protection for influenza. We have a program for coronavirus and for HIV using the same platform. And there's a bunch of other pathogens we could apply it to. The big things that have happened in the last 8 months was that there was this massive de-risking during the pandemic of the mRNA LNP vaccine delivery platform, where instead of delivering protein, you deliver RNA instructions, those get into your cells, and then you make like farm fresh protein right there in situ in your arm and your lymph node. And I always thought that platform would be very attractive to us because we're inherently a high valency vaccine, right? We have 18 components for our flu vaccine. That is a pain the first time you manufacture it. Once you've built it, you don't have to ever build it again. You can use the same thing. You can manufacture it and stockpile it every year. You can dominate those markets because you're not having to make a new vaccine in the northern and southern hemisphere every year. But the first time you make it, it sucks. And the mRNA technology radically simplified the challenge of creating 18 components. And so I'd watched that technology for years, but it had, technology readiness questions. And the pandemic changed that. There suddenly was billions of people who had received the vaccine. So we know the tolerability and safety profile, the efficacy of the vaccine. And then second was a cottage industry of a whole bunch of companies that now offer contract research organization GMP manufacturing services, which didn't previously exist. And that was a second prerequisite. The technology needed to be safe and de-risked, but second, the FDA needed to like it. But then third, I needed somewhere I could go manufacture the damn thing because we didn't have that in-house expertise, and GMP is its own beast. And so the stars really aligned. So we went all in on converting our lead program that we were getting ready to initiate GMP. And I was like, it's protein. And I said, let's hold off. Let's see if we can run it as RNA. And so we ran all those studies and it looked phenomenal. If anything, the titers are much higher now, which was one of the features of RNA. It has kind of a very attractive adjuvant-like property that produces very high titers. And so we've put it into mice and ferrets. We've got a pig study geared up. And it produces this ultra broad spectrum response. And the consequences of that for Sinovac were pretty huge. On the partnering side, it opened up a whole bunch of the top 10 vaccine pharmas that were interested in talking with us. We were already in pretty deep diligence with one group that was interested in protein anyway. They were also interested in RNA. And they're putting together a partnership and collaboration proposal. And then since we had the RNA data come through, then suddenly I think it's 9 of the top 10 vaccine pharma we're all talking to and in discussions around partnership. And so there's just been a phenomenal expansion of interest. And I think it's because they either have built up infrastructure and a war chest because of mRNA, or they're going into mRNA. And they can see us as, hey, we can take our mRNA platform and investment and we can plug and play Sinovac's universality. So we can make those vaccines universal, which opens up the platforms to a whole bunch of pathogens that either are poorly addressed or not addressed at all without. And so that's been a huge game changer for us. Even the vet space, we've had a big expansion. So we're talking to 3 main vet groups. One of them's putting together a study that they would validate. We'd send over the vaccine, they'd run it at a university they'd pay for the study. And we're looking to license it out for veterinary application as we move towards humans. And that's also led to our Series A. So I think maybe I said last time we're aiming to close in October. I think we're actually a little ahead of schedule, so maybe more like September. But we're having a great Series A, and the RNA enabled us to have a much cheaper Series A. It was a third of the cost to manufacture with mRNA LNP, and it took up almost half the time. And so it was, for a company, that's a big deal, right? Because we have a burn rate. So being able to get in clinic, I think right now we could start in clinic in 2024 and we would finish in Q1 of 2025 of our phase 1 major inflection point. And that was just not possible with protein. And so I think those have been major advances for us as a company. The Series A has been, I think worlds different than fundraising in 2022, where I think everyone was just freaked out about the economy. And I think also our technology has matured and we're right in the zeitgeist of this huge cheerleading. I think there is some cheerleading hype going on around mRNA, but it also has some very concrete, useful things. With the 3 that I mentioned, cheaper, faster, higher titers, those are all key. But it also enables you to do really cool computational immunoengineering of vaccine responses in a way that is not possible with protein. And so I think we're also looking toward the future of us being able to innovate on the platform that's attractive to us. So those are the major updates. And it's a good time to be in vaccines.
Great. So as we were talking before we got started here, Jake, I just came from a conference at Brigham Women's, and I'm reminded kind of what one of the speakers said. He said, we can certainly cure cancer in mice. The problem is when you try and recreate that in humans. The physiology just isn't the same. Are there, as you now think about going into the clinic, are there challenges that you already anticipate, for the vaccine?
So I agree. It's the reason that I did the hard work, and I didn't work with mice in the beginning, right? Mice are the easiest thing to work with, but they're, excuse my French, they're a shitty model for a lot of diseases. And they don't really get the flu. They have a couple kind of partially adapted artificial situations. And it's the reason I'm an affiliate professor of the University of San Carlos in Guatemala, where I grew up. It's the reason really my brother, in coordination with me, we built a facility in Guatemala and worked with collaborators down there to run studies in pigs, full-grown pigs, outbred pigs, rather than in mice, because pigs actually get the flu. It's a $185 million per year market for flu shots for pigs. The same pig strains end up infecting humans. And pigs are the recombination species that causes the zoonotic pandemic shuffling of avian and pig and human strains. So I worked on the harder model, the bigger model, but the much more valuable model. And that is always my philosophy. And cancer, for instance, those guys should be working with dogs. Dogs actually get cancer, real cancers that cook up over years. They're not like a mouse fake cancer. I think if we did more work like that, we'd have more useful drugs advancing. And so I did the hard work with pigs. And that's where we developed a lot of the data. Then we expanded into ferrets, which is a big model. It's important. We see the same effect. And then we actually did mice more recently as a tool, because we have a facility here that enables us to do lots of rapid compatibility testing. But again, mouse is not a great model. It's easy, and the easy is not always the right answer. So that's what we've done so far. We have 3 organisms that are all showing us the same story. We're also looking at these human immune organoids with a company called Parallel Bio. That they take lymph nodes from people that are donors. They kind of break them up and create these little fake lymph nodes you can immunize. And so it's the closest thing before getting into a human to kind of testing your vaccine in a human. So we're doing that. It's still, like you said, it's a quantum leap. The first time you put it in a human, it's different. And it's a new organism. I'm going in pretty confident because 3 organisms are all telling the same story, and they're very genetically distinct. The difference with humans is they live a lot longer, and they've been exposed to influenza and vaccines a lot more times than these animals. So lots of the studies we've done are on naive animals. These are animals kind of like they represent a child, where they haven't been exposed to flu or the flu vaccine before. And that means that they don't have a bunch of B cell memory against historical strains that might get in the way of producing a novel response. But we've also run imprinting studies where we took pigs and we gave them a whole bunch of like a 2007 shot, and then gave them 1 shot of our vaccine to see, are they going to re-elicit 2007, which is the problem with imprinting, or are you going to create a big breadth? And we saw the big breadth. We're actively running studies on ferrets and pigs where we've infected them with historical strains, and in some cases vaccinated them with historical vaccines. And so we're going all in on this because we think that the imprinting issue is a key advantage of us, that our technology, it doesn't matter if you have imprinting against historical strains, because there's not enough of that strain in the vaccine, even if that strain's in the vaccine, for you to produce a focused immune response against it. You still only produce a response for B cells that have recognized the shared sites. And so we're running, this will be our 3rd studies addressing that. And that's, of course, something we want to look at in the humans. The other thing is that elderly populations, historically behave different. So once people are, let's say, over 60, there's a process of immunosenescence that begins where the immune system just gets a little more sluggish to respond to vaccines. This is a known issue for flu vaccines. It's been historically dealt with by increasing the dose. And we have a dedicated the phase 2 cohort, a phase 2 arm that addresses the performance of the vaccine and explores a couple doses in the elderly population as opposed to the full adult, and then finally the pediatric. So these are the things we'll check on. The last detail is geographical differences. So it's been known in some vaccine studies that they ran a vaccine was run one time in the UK, and it behaved a certain way. And then when they brought it over to Africa, it behaved differently in that population. And so testing at lots of different global sites matters early to make sure you have in the same way I chose pigs rather than mice, I think you need to do the right choice rather than the easy choice and pick lots of diverse geographic locations for your vaccine. And ask the real question, how's this going to perform in the world? So you don't end up saying, well, it looked great in the UK and I'm surprised that it underperformed elsewhere. And so those are the things we're going to go do to test it. I think we're sitting on a remarkable body of broad spectrum data we've built up, and the LNP RNA vaccines, we kind of know how those have performed in global populations in terms of immune stimulation. It was pretty standardized. And so I'm not too worried about any one thing, but those are the things we look at.
How many people do you think will be participants in that, large FDA study?
Last I checked, I think it's about 3,000. I will go double check on those numbers. I think that was it. These are pretty straightforward studies. They're outpatient procedures. You get a vaccine, you take some blood there, you take it later at a couple points. I think the number is about 3,000. I would need to go double check that to get you the exact number. And I think that's really still a subject for discussion right now.
Right. Okay. Well, it sounds like you're really moving along, Jake. I'm very happy and I guess proud of the work that you've done, because I think it's just such a great thing for the world, and so representative of all of the change that's taking place in biotech. So thank you very much for your time, and I look forward to following up again with you.
Oh, thank you, and it's always great talking with you.
Let's see. I hope that these short segments have given you some feeling for the pace of the strides being made in healthcare innovation. Yes, our founders faced challenges as they raced to bring their systems and therapies to patients. In the most extreme example, Rasab was adapted by taking the dramatic step of moving his operation to Malaysia. By now, I think everyone on the planet has heard of ChatGPT, generative artificial intelligence. And we have some feel for the quickening pace of innovation that it will engender. A few months ago, we spoke with Ian Strug at Virgo, which is using AI to better and more quickly populate clinical trials. That alone may cut the time involved in drug testing by perhaps 50%. Think for a minute about what that might do to the cost of bringing a drug to market, and therefore how it might reduce the prices patients ultimately pay. Jake Glanville has used data and computational biology to construct a universal flu vaccine, which, if efficacious, could save hundreds of thousands of lives a year. But aside from the productivity which would come from saving all those lives, think about how many man-hours of productivity can be saved simply by reducing worker sick days. Data and the AI which brings it to life has the capacity not only to do the obvious, to refine physician and hospital workflows, but also to impact patients directly by improving traditional therapies that already exist. For example, a study of over 2,100 women in Norway with early stage breast cancer found that gene expression testing would change therapy decisions in almost 1/3 of cases, with 45% of those assigned chemotherapy prior to the testing being de-escalated to endocrine therapy following the results. All of that only scratches the surface of the good news. But the news isn't all good, because in order for these innovations to have their full effect, healthcare systems will have to change. Tan's issues were not with his tech. They were about the sometimes invisible roadblocks in systems in which he tried to apply that tech. And as is so often the case, government has to stop politicizing issues which upon further examination contradict their policy goals. The FTC recently shocked the biotech world by opposing the acquisition of Horizon Therapeutics by Amgen on the premise that it would give more pricing power to Amgen in the bundling of other products. But the perennial acquisition of promising therapeutics produced by small biotechs, dedicated only to the science, has always been the way the industry has financed itself. Risky drugs are built and initially tested in small companies. Then, at either the expensive third and final testing stage or immediately after, when sales and distribution become paramount, the smaller companies are absorbed by larger companies focused more on business than science. As I said at the start, the wonderful thing about the people doing the science is that they don't care about money. They understand science, but they're not in the least interested in learning how to sell and distribute the therapeutics they produce. The people doing the science in these small companies make tremendous personal sacrifices, not only in terms of their wealth, but in terms of family and other personal relationships as well. AB talked about this. But if government must reconsider policies which conflict with their goals, some founders may need to reconsider their own proclivities as well. I started this podcast today with my expression of support, indeed my own inspiration, coming from their selfless pursuit of progress for patients. In preparation for my first interview with Jake Glanville last year, I read a fabulous book by his CFO, Stephanie Wisner, called Building Back to Biotech, about applying basic business concepts to the building of a successful life sciences enterprise. At that time, I was surprised that the book even had to be written. Today, however, I can understand why. I'm now reading a book recommended by Rasab called The Emperor of All Maladies, about the history of cancer. One interesting part of the book explains how Sidney Farber recognized the importance of public relations and marketing in the rallying of public opinion to raise funds needed to continue his early battle against childhood leukemia through the creation of his Jimmy Fund. Though business concepts and measurements may seem pedantic to some life sciences founders, they're an important component to actually performing the science, especially in a new environment where money is no longer free. Next month, we'll talk with a new biotech founder whose grit and determination will inspire even the most skeptical among us. Until then, thank you for joining us for today's podcast. We'll look forward to seeing you again next month.